I'm going to call upon each of the speakers, the first being Barbara Imperiali, who's the class of 1922 professor of chemistry and biology at MIT. She's interested in protein structure, function, and design involving synthesis, and we very much look forward to her presentation. Sure.
Not 1922.
Not 1922.
It is the class of 9, actually. Okay, so I'm going to kick off these sessions with a somewhat biochemical talk to discuss with you our progress with developing activity-based sensors for protein kinases, because they play such a fundamental and central role to signaling processes which so often go awry in cancers. So the sensor attributes that I'll present to you concern fluorescence-based sensors that we have a non-radioactive that can be adapted with very simple design rules to all the common types of protein kinases, and that they provide with direct continuous activity assays, in contrast to a lot of the current approaches, things like Luminex or Luminex, mass spec, or other antibody-based approaches, which really often look at surrogates for activity, are commonly discontinuous, and are deeply reliant upon the availability of high-quality antibodies. The sorts of assays I'll talk to you about are in the solution state, but we hope to be able to parlay these forward to more bead-based assays to really give them a high practicality. But at the current time, the types of sensors we make are very useful in high-throughput screens for small molecule drug development up to the 1536 well plate format and beyond. Most importantly, these sensors employ physiological ATP, which is critical, and the sensors we've developed in the past are available under the Omnia trademark at Invitrogen. Protein kinases often recognize linear sequences of peptides and make recognition elements with the N and C termini of a short oligopeptide embedded within the substrate for the kinase. And those are very simple to adapt to the quinoline-based sensing method that I'll describe to you. However, there are other kinases that are really critical in cell biology, in particular the MAP kinases, that employ an extended recognition mode. And if we are to present to the community sensors that can be applied in crude cell or tissue lysates, we need sensors of the optimum selectivity in order to be able to use them directly on really small samples of material under unfractionated conditions, because as soon as you start fractionating enzymes, they act up on you and lose activity. So the design principles I'll describe to you for a set of MAP kinase sensors, in particular JUNC, ERK, and P38 alpha, are based on a principle that employs a small modular docking domain within the sensor structure, a variable linker, and a motif which becomes phosphorylated, but upon phosphorylation, a fluorescent signal is generated because embedded within that phosphorylation motif is a quinoline-containing amino acid developed in my group that lights up upon transfer of a phosphoryl group to the phosphorylation site, complexation with magnesium, which then causes chelation-enhanced fluorescence. Each of these modules can be, in a very satisfying way, be independently varied to drive towards very selective sensors. The basis for the signaling is well established now in the group and is a very powerful signal because there's no background in the absence of the chelation effect. So for JUNC sensor optimization, which was one of the challenging ones, we had not been able to achieve good selective sensors because JUNC kinase recognizes a very short recognition motif, just a threonine proline type motif within the local sequence that becomes phosphorylated. So we were attracted by the ability to generate a small docking domain which would bind to the kinase, and there's a lot of literature about these docking domains. They are at variable distances from the phosphorylation catalytic machinery, but they can be optimized, and we can optimize these through standard biophysical methods. We can then look at short peptide sequences and optimize those through just using the activity assay, but not in the presence of complex mixtures of cell lysates. And then we can further optimize the variable linker between them through flexible or less flexible motifs to drive down the affinities of the sensor, of the kinase for the sensor, into the very low micromolar range and achieve high maximal turnovers, and ultimately get a very selective sensor. The criteria by which I describe these sensors, I just want to show you what we do in order to benchmark against well-known mechanisms. The first thing one does is just look for activity and turnover within pure recombinant enzymes. And you can see from this panel of MAP kinases and other related enzymes that the sensor that I described, shown in black, has a very strong fluorescence activity. This is a real-time readout of activity. It's not a quench point, it's not the 60-minute point or the 1-hour point. This is real activity. We can then treat the cells, MCF7 cells in this case, with a stimulant, in this case anisomycin, and watch for the progression of junk activity over a time course. And this correlates beautifully with the traditionally applied Western blot methods that look at phosphorylation-sensitive antibodies recognizing the activated form of junk. This is a little confusing because of the splice variants of J and K that are known in the literature. This is useful for junk 1, 2, and 3. They don't differentiate very much. Some are tissue differentiated, but nevertheless, this is a global assay of junk in this case. These cells can also be treated over a time course with small amounts of very, very selective junk inhibitors. These are the types that my colleague Nathaniel Gray has developed, and we can see very clearly that pre-treatment with the inhibitor systematically will depress the activity way down to a background level. And once, sorry, my finger, my finger's trigger finger indeed. And we can really see the activity depressed. And on this side of the graph, you will see the IC50 for the junk inhibitor against junk in crude cell lysates as well as pure recombinant enzymes. And these recapitulate very, very well, showing us that our sensor, what we are reporting with the sensor, is really junk activity in the crude unfractionated cell lysates, rather than a bunch of other different kinases. And this is what we generally do to validate sensors. We have additionally exploited this design principle for the development of ERK and P38 alpha sensors. And this is an example of our results with the ERK sensor, once again showing very good repression of activity in the presence of an ERK selective inhibitor. And while in high levels of recombinant enzyme, we see some crosstalk to P38 alpha, when we carry out the studies in the presence of the P38 alpha inhibitor, we very faithfully reflect the ERK activity in the crude unfractionated cell lysates. So to profile MAP kinase activities, we can literally take very small amounts of cells, treat them with very different types of conditions, and then go in and profile the activities of each of these kinases, in addition to a set of others that are amenable to a very simple design principle, and really get very exquisite data on the activity enhancement of certain kinases over a time course perhaps. But what we're doing currently with the YAFI group is really to look to see how certain cell lines, cancerous cell lines, respond to different drug treatments, and profile the kinase activities within those cell types to really assess the pulse of the cell with respect to these signaling nodes. So here I show you a slide that is one from a paper by Doug Loffenberger and Mike Yaffe, and I point to you very specifically to the kinases shown in red, ERK, JUNK, and P38. And what I want to stress by this slide is that the sensors we'd made in the past were not sufficiently selective to any of these kinases to really be able to interrogate crude tissue or cell lysate. But the current family of MAP kinase sensors really hits this set of signaling nodes that are very important for really understanding the pathway. For tissue lysates, we've shown very clearly you can take crude samples of tissue, lysate, spin them down, and once again get sufficient activity to actually look at those, profile those kinases in crude unfractionated diseased tissue lysates. And I believe, yes, that is it.
Thank you. We take 2 questions if we could, something like that. Yep, please.
Yes.
Great talk. Have you looked at this in actual tumor lysates as opposed to cell lines?
Yes, we, due to sort of limited access to these at the time, but maybe an event such as this might stimulate, we, from the NDRI, acquired tissue lysates from healthy and disease-adjacent cancers, and we looked at certain breast cancer samples, lung and prostate cancer samples, and we saw for a set of kinases, AKT, ERK, P38 alpha, and PKA, clear differentials of the activity of the kinases in those tissues. Those samples were all run in 9, not just triplicate, but 3 times triplicate of the samples. We got unbelievably robust measurements of the activities of kinases and could profile distinct changes in the crude tissue lysates. Yes.
Questions, please. I have one. Could this be used in organs? I mean, could you arm an organ to express one of these things? I mean, you can't express it biologically, but can you? Is there a chemical trick one could use to make an organ take such a small molecule up?
Not quite, because our wavelengths are not good for the visualization under those conditions. They would need to be in the much longer, I would say, IR detectable region. But the principle could potentially be employed if we then sort of further arm the fluorophore to bring it right to those wavelengths to enable that. So use chelation-enhanced fluorescence, but with something that delivers a longer wavelength emission.
Signal transducer to
Yeah,
another reporter.
exactly.
I mean,
So
that'd be very
It
serious.
would be very exciting, yes.
Yeah, because, you know, one of the questions always is, in making PD measurements, does the drug actually hit the target? And, you know, we do all kinds of crude assays on dead cells that are FFP fixed and whatever. Wouldn't
Right.
it be nice to do it in real life? Okay, are there others? If not, we want to thank Dr. Imperiali.